Hello students, good morning. I am Sathiyaraj Srinivasn, and in this presentation we are going to see about the radiation resistant microorganism, especially the bacteria which are showing the resistance to the radiation. The radioactive pollution occurs when there is a presence or the deposition of the radioactive materials in the atmosphere or in the environment, especially when their presence is accidental and when it's present in the environmental threat due to the radioactive decay. The destruction caused by the radioactive material is because of the emission of the hazardous ionizing radiation. So they will produce like alpha, beta particles and also gamma rays, sometimes the neurons in the environment. The radioactive substances are characterized by the radiation because there is a lot of instability in the particles present in the radioactive materials. The radioactive materials can seriously affect, alter, even destroy plant, animals and human life. The extent of the damage or danger to the environment depends on the radioactive material concentration and the energy emitted by the radioactive materials and also the radiation type. So in this presentation we are going to see the detailed explanation of how the radiation cause the damage to the microorganisms and what are the effect of the radiation and how the radiation resistant bacteria are useful for our biotechnological application. So we are going to see each slide with explanation and the details. There are two kinds of radiation is available. The first one is the ionizing radiation and second one is the non-ionizing radiation. Ionizing radiation include X-rays and gamma rays, and non-ionizing radiation include UV. For several years we use X-rays and UV for oxidative stress response studies. And when we use the ionizing radiation, the reactive oxygen species, that is ROS, are mainly produced inside the cell. So those ROS are used to investigate the nature of mutagenesis and disease, aging and finally the cell death. So we use the ROS to study the mechanism of the cell mutagenesis and other cell aging and cell death studies. For several years we thought that the DNA damage is the universal criteria for the cell death. During the irradiation, especially the ionizing radiation by the gamma rays, the DNA is mainly damaged. So this is the classical radiation toxicity model. So we thought that the DNA damage is the major cause for the cell death. So the next slide is going to show you the recent studies how we change from DNA damage to other kind of toxicity by gamma radiation. For several years we thought that the gamma radiation or the non-ionizing radiation which produce the ROS are targeting the DNA. So the DNA is the major target for the ROS and the radiation which cause the damage to the cellular model. So several classical toxicity model mainly target the DNA as the universal target. But the recent studies in mammalian and prokaryotes are find out that the proteins are the initial target of the radiation. So when the radiation affect the cell, even the mammalian cell or the prokaryotic cells, so the proteins are oxidized first and which cause damages to the whole cell and also which leads to the cell death. So several organisms, especially the several different kind of bacteria, we have used to study how the cellular damage by the radiation. So during the studies, we found out that several kind of proteins, especially the enzymes, are initial target for the ROS produced by the radiation. So several enzymes are damaged very severely by oxidative damage, and which leads to the inactivation of the enzymes. And because of inactivation, the cell cannot able to repair the damages, and finally they leads to cell death. So this slide showing how the radiation pollution are spreading across the earth in various regions. So for example, the underground waste disposal are usually the source of the radiation and which are mixed with the groundwater. So the groundwater move into the lake and go to the all living organism in the lake. And when the lake is evaporating, they are moving to the atmosphere. And during the rain, the radioactive materials are coming back to the ground and contaminate the entire ground. So the radioactive materials present in the waste disposal site are very dangerous, which are the main source of the radioactive problem all over the world. And second most problem is the uranium mining mines. So those minings are producing several radioactive nuclei which are going to the atmosphere, and from the atmosphere they come back to the earth by the rain, and then they go to the plant and mix with the other living organisms. So when we study about the radiation target, so most papers are nowadays targeting and also demonstrated the critical target of the radiation is the protein. Because when we study the DNA damages during the radiation study, the damage of the DNA is very similar in both radiation sensitive and radiation resistant bacteria. So that means the radiation affect similarly to both radiation sensitive and also resistant bacteria and cause similar kind of effect to the DNA. But in case of the radiation resistant bacteria, the proteins are highly protected from the radiation effect, and they are not oxidized like a radiation sensitive bacteria. So that means the radiation resistant bacteria contain several proteins which are very resistant to radiation. When we talk about the radiation resistant bacteria, everybody know that the Deinococcus is the one of the popular genus which have the radiation resistant bacterial species. So especially the Deinococcus radiodurans is very popular model for the radiation resistant bacteria. So Deinococcus radiodurans survive with a 12,000 gyres. So 12,000 gyres means around 12 kilogyres of the X-ray or gamma rays. So this is almost 20 times greater than the Escherichia coli. So Escherichia coli is the one of the model organism for the microbiological studies. So by comparing the E. coli and the Deinococcus, we can see that how much the Deinococcus can resist the radiation. And when we compare with the human cells and Deinococcus can resist almost 3,000 times compared with the Deinococcus radiodurans. That means the human cells can be destroyed even with a very low dose of radiation. And also Deinococcus can survive with a long time with very low concentration of the humidity. That means they are very drought resistant. They can survive even with 5% of the water, and they can survive for more than 2 years. So this slide showing the survival curve of the different kind of organisms comparison. So in this graph you can see that the human lung fibroblast with around 3 gyres. So here is around 3 gyres. They are making around 72 double-stranded breaks to the DNA. But if you see the Shivanella, this is the bacteria, is making around 1 double-stranded break with less than 100 gyres. But in case of the Deinococcus, with 10,000 gyres, they make 160 double-stranded breaks. So by comparing the human and the Deinococcus deridurans, the dose of the gamma radiation is very low, but even the low concentration makes several double-stranded breaks and which lead to the cell death. So here is the diagram to show how the DNA will breaking with the DNA damage to the radiation. So when the radiation affect the cell contain the DNA. So when the DNA is affected by the radiation, they make a single stranded break. That means one strand of the double stranded DNA will be break and cause the DNA to inactive. And also they make a double stranded breaks. That means totally the DNA will be broken down into 2 pieces. So likewise they completely destroy the DNA. So with broken DNA, the other function of the cell will be controlled. So the cell cannot produce any metabolism and they cannot able to survive the radiation. So that's the major reason the cell will die. So in this figure, you can see the comparison of the radiation resistant bacteria and radiation sensitive bacteria, or how they are affected by the ionizing radiation. So this direction is the way how the radiation are causing the cellular chemical changes inside the cell, and the top one is the sensitive cell and the down one is a resistant cell. In both cases, the ionizing radiation cause the indirect effect and make the double stranded breaks. And similarly, they produce the hydroxyl radical and hydrogen peroxide. So the reactive oxygen species is causing the uniformly distributed cellular damages to the sensitive cells. But in case of the resistant cells, the antioxidant metabolites complexes which contain the Mn2 plus, that is the magnesium Mn2 plus complex, are very interact with the reactive oxygen species and reduce the damages to the cellular proteins. But in case of the sensitive cells, they cause the very specific protein damages, and due to the several damages to the protein and also the double stranded breaks in the DNA, they will lead to the cell death by the protein damage. So because of the protein damage, the DNA repair will be malfunction and they cannot repair the DNA, and that means the broken DNA due to the double stranded breaks are still present in the cell and which cause the cell to die. But in case of the resistant cells, due to the antioxidant metabolite complexes, they will protect the protein from the oxidation, and the protected proteins will repair the genome, and the genome reconstruction will occur inside the resistant cells and the cell will survive. But in case of the sensitive cell, the DNA, the chromosome is broken to the several pieces due to the double-stranded breaks, and which lead to the cell death. So the sensitive cell will die, but the resistant cell will survive after the radiation. So this figure shows the phylogenetic distribution of the radiation resistant organism. Among the bacteria, the blue color diagram, blue color branches, you can see that Deinococcus radiodurans and the Dermis dermophilus are the two core genus which evolved from the common ancestor and form the radiation resistant clade only in the Deinococcus. By comparing the Deinococcus, Dermis dermophilus doesn't have the radiation resistant, but they are all started from the common ancestor. And similarly, if you see the eukarya, and some metazoa have the radiation resistance, and in case of archaea, some halobacteria species have the radiation resistance. So by comparing the overall phylogenetic tree, we can understand that the radiation resistant species are evolved independently from each ancestor and accumulate several genes which can make the bacteria or the organism to resistance against radiation. This is the overview of the isolation protocol to isolate radiation resistant microorganisms from the sources. The first step, we collect the soil sample from environment. Probably the environment doesn't have any radiation, but this is the just normal soil samples. And after collecting the soil samples, we treat the soil with gamma radiation. For gamma radiation, we use the cobalt 60 gamma irradiator, and after irradiating the soil sample with various doses of gamma radiation, we serially dilute the soil sample using the saline. So after serial dilution, we spread the serial dilution to the agar plates. We use usually the R2A agar plates, or we use different kind of agar plates to isolate various microorganisms. So once we found the colonies, and we can pick the colonies and transfer to the new plate for pure culturing. So sometimes the pure culturing take 1 or 2 passages, and once we found the colonies are very pure, and we go for the further identification. So here is the table of bacteria which are isolated in the year 2020 after radiating the soil sample with the gamma radiation. So you can see that several bacterial genus are present after radiation, but the major genus are both Hymenobacter and Deinococcus. So Hymenobacter, there are 59 strains, and Deinococcus 28 strains. And the highlighted one, the blue color highlights, they are the bacteria or the bacterial isolates which have the very low similarity, that is less than 98.7% similarity with the type strains of the same genus. So you can see that there are several new species we can see from the Deinococcus, Hymenobacter and Microvirga, Rufibacter and Spirosoma. So previously there are several articles describing only the Deinococcus is the radiation resistant bacteria, but in our study we found out Hymenobacter also one of the popular genera containing similar kind of radiation resistant bacteria like Deinococcus, and also the Microvirga also showing the radiation resistance, but the resistant level is lesser than the Deinococcus or Hymenobacter. But the Spirosoma and Rufibacter showing the similar kind of radiation resistance, similar like a Deinococcus and Hymenobacter. So in our study we found out not only Deinococcus, the Hymenobacter and the Rufibacter and the Spirosoma are all the dominant genera which have the radiation resistant microorganisms. So overall in this study we found out around 135 strains which are new species isolated from the soil sample. So here is the phylogenetic tree of the Deinococcus. So you can see that this is the Deinococcus deriodurans. This is a type strain of the Deinococcus, which is as a popular model for the radiation resistance. We have isolated several Deinococcus which are very close with the Deinococcus radiodurans. So they show very high similar radiation resistance value, similar like a Deinococcus radiodurans. And similarly, here is the phylogenetic tree of the Hymenobacter genus. There are several Hymenobacter genus reported from the laboratory which have highly similar radioactive resistance level like a Deinococcus. So you can see the popular one is the Hymenobacter svenensis and Hymenobacter humi and Hymenobacter terrae. So they are very resistant to gamma radiation. And this is the phylogenetic tree which shows the gamma radiation resistant Spirosoma. So the Spirosoma pulveris and radio tolerance and svenensis and Panacea, they are all isolated and reported from our laboratory. So this is very interesting to see. There are several different bacteria from different genera can also show the similar kind of radiation resistance, similar like Deinococcus model. So the next slide, we can see the location of the each genera in the overall view of the Deinococcus. In case of the Deinococcus, they are present in the Deinococcus dermis phylum. But in case of the Microvirga, they are belong to the alpha proteobacteria. But in case of the Hymenobacter and the Spirosoma, they are belong to the Bacteroides phylum. So they are totally belong to the different phylum. For example, Bacteroides are here, and the proteobacteria, the alpha proteobacteria are present here, and Deinococcus are here. So that means the radiation resistant genes are spread across the different level of phylum in the bacterial kingdom. So that means there are several different bacteria. They have no relationship with each other. They doesn't have the close relation with each other, but they have the genes which can show the radiation resistance similar like a Deinococcus. That means those bacteria are evolved individually by accumulating genes which are present in their environment. So the evolution of the radiation resistant bacteria, we cannot say that they are really related each other. So that means the genes are possibly are occurred or accumulated from the nearby environment, and they are evolved by different level of evolution. The gamma radiation resistant bacteria also show the resistance against the UV radiation. So here you can see the comparison of the Deinococcus radiodurans with Hymenobacter and E. coli. The E. coli cell will die or cannot survive even with 100 joule of the UV flux, but the Deinococcus and the Hymenobacter can survive even very high level of radiation, UV radiation. So you can see the log10 value, the survival rate is around 10% even after 1200 joule meter square of the UV radiation flux. So you can compare the relationship between the survival and the exposure to ionizing radiation in this figure. So the first figure show the gamma radiation intensities with 0 to 18 kilogirays and the log survival ratio. So 0 means 100% survival, and minus 1 is a 10% survival, and 0.1% and 0.00% survival. So with even the low dose of the gamma radiation, the Shivernella and Neisseria and Pseudomonas and E. coli and Dermis, they all cannot able to survive even from 0 to 2 kilogirays. But if you compare the Deinococcus geothermalis and the Deinococcus radiodurans, they can able to survive the 16 kilogirays, 16 to 18 kilogirays of the acute gamma radiation. And similarly, they have here this a comparison of the intracellular manganese ion ratio with the survival rate, similar like the figure A. So when the manganese ratio is very high, like Deinococcus radiodurans and geothermalis, the bacteria can able to survive with a higher radiation rate. But when the concentration is very low, like Pseudomonas putida, and the Shivernella oriensis and Neisseria gonorrhoeae and the E. coli, when the concentration is very low, they cannot even to survive the low concentration of the radiation. That means the intracellular manganese ion ratio is one of the key factor for the cell survival, because the manganese ions are very important to control or reduce the oxidation effect of the protein when there is a radiation. So this graph is showing the survival fraction or survival rate of various Hymenobacter and Spirosoma species. So those are all the Hymenobacter species, and those are are different Spirosoma species, and the R1 is the positive control, that is a Deinococcus strain. So you can see that several bacterial species in the Hymenobacter and the Spirosoma show similar survival curve like a Deinococcus. So this experiment actually done using the TYG broth, using the stationary phase cell as an initial cell for the experiment. So we use the stationary phase cells, and we give the gamma radiation, and then we serially dilute the diluents, and then we plate them in the TYG plate, and after 2 days of incubation at the optimum condition, we will count the colonies and make this graph. And similarly, this is the same strains by treated with the UV radiation. So similar like the Deinococcus radiodurans, several Hymenobacter and the Spirosoma strains showing the resistance survival after the UV radiation. And here is the comparison between the human and the human cells versus the Deinococcus radiodurans. So this experiment is we can see how much is the radiation affecting the human cells. So even the low dose of the radiation, like a 10 Guy rays, human cell cannot able to survive. But in case of the Deinococcus radiodurans, they can able to survive even more than 10,000 Guy rays of the radiation. So you can understand the difference, how much the Deinococcus can able to resist the radiation. So if we want to understand how we can understand the mechanism of the survival of the radiodurans, we can study the genome of the radiodurans and compare the genome and compare the other mammalian genomes to study the difference between the protein and the metabolites of the Deinococcus and the other higher organisms, to confirm which are the genes are involved with the radiation resistance. So the next slide, we are going to see the unique domains of the Deinococcus species, which shows very important role or key role in the radiation resistance. There are several articles are published which shows the unique domain architecture of the Deinococcus proteins. So the most popular one in the study, the DNA helicase superfamily, and Mercae like nuclease, and hydrolase, and monoxygenase, and Recae or Recae like genes, Recae proteins. They are the major domains which are very important for the Deinococcus to survive after gamma radiation. And Deinococcus, especially several different Deinococcus and also the Deinococcus radiodurans, are used for the bioremediation of the environment. Especially the Deinococcus radiodurans can able to survive with a high level of the radiation. When there is a radiation and also some other kind of contamination in the environment, the normal bacteria or normal microorganism cannot able to survive in the toxic waste environment. Only the Deinococcus or the radiation resistant bacteria can able to survive, and also can able to utilize those toxic waste for the growth, and then they can able to remove the contaminant from the environment. But in case of the normal other bacteria, which are killed by the radiation toxicity, but only the Deinococcus or other radiation resistant bacteria can survive and utilize the both metallic and organic contamination from the environment. And here is the picture of the Deinococcus radiodurans. You can see that they have the large tetrad forming bacteria. So you can see there are 4 bacteria are combined together. So and also you can see that they have very clear nucleus, and each bacteria contain multiple copies of the circular chromosome or the nucleic material, so that they can able to resist the radiation. And here you can see a comparison between the resistant and non-resistant bacteria with gamma radiation. In case of the control, all the bacteria are surviving. But when we apply the gamma radiation, the Shigella and Escherichia coli and the Pseudomonas are die. They are not surviving. And when there is a Deinococcus radiodurans with REC 30, that is a knockout mutant, that is without the REC-A gene, they will not able to survive, because REC-A is one of the important protein for the cell survival after gamma radiation. So Deinococcus survive with the one of the key enzyme REC-A. And we have also the comparison with the metal remediation with the mercury resistance and gamma radiation together. So this is the control which apply no radiation, but you can see that all the bacteria can able to survive without the radiation. And this one is the gamma radiated plate, which have the normal bacteria, normal Deinococcus radiodurans, which have no mercury resistant gene, so they will not survive. This R1 doesn't have the mercury gene, and similarly this bacteria have the mercury gene but doesn't have the REC-A, so they will not able to survive. And E. coli cannot able to survive because they doesn't have both radiation resistance and also mercury resistance. So all these bacteria or bacterial strains cannot able to survive because either they lack the REC-A or the mercury resistant MRA. But this is the Deinococcus radiodurans with different copy numbers of MER-A gene. So MER-A gene actually producing the mercury resistance. Okay, so MER-A gene 150 copy and 20 copy and 1 copy and 10 copy. So this all different copy numbers of the MER-A gene containing Deinococcus can able to survive and remove the mercury from the media. So this is the confirmation that the Deinococcus radiodurans strains which expressing the MER-A during the growth at 6000 rad per hour of radiation can able to resist both radiation and also the mercury in the media. So we can utilize such a bacteria for the remediation of the mercury from the environment, which also contain radiation in the environment. So the genetically engineered bacteria, the Deinococcus, can be used for the bioremediation to digest solvent and heavy metals even in the highly radioactive active sites. In case of the normal bacteria, even the bacteria such as the Escherichia coli cannot able to survive even if they have the mercury resistant gene. but the Deinococcus can able to detoxify the ionic mercury, because they can able to resist the mercury and also can able to resist the radiation. And also we found that some of the radiation resistant bacteria can able to wound healing effect. So one of the Deinococcus, the BM2 strain, that's the Deinococcus actinodurans, which was isolated from the Korean soil, which shows almost the similar radiation survival rate compared with the Deinococcus radiodurans. So you can see the survival curve is almost similar with the Deinococcus radiodurans, which shows really good effect with wound healing activity of the BM2 strain, the extract from the BM2 strain with the arcade cell. So this is the control, and this is the BM2 extract 10 microgram per ml, and this is the BM2 extract 30 microgram per ml, and 0 time, and 12 hours and 24 hours. So by comparing the control and 10 microgram and 30 microgram, you can see that the wound healing activity of the BM2 strain is very significant, and they produce very very good wound healing activity by comparison. So likewise, we can utilize the radiation resistant bacteria for several bioremediation activity and also some medicinal properties. So I hope that it's very interesting to study about the radiation resistant bacteria, and we will continue to study the radiation resistant bacteria and their mechanism. And thank you for your listening. If you have any questions, you can ask me by my Kakao message, or you can contact me in my office.